welkom, ons is baie opgewonde om julle te hê. En ons is baie opgewonde om vir julle te hê. Rick, thank you very much for taking the time and all the effort to come over here. Ons sien almal uit na 'n lekker gesprek vanoggend, so please feel free to ask some questions, give your perspectives, your opinions and so, and enjoy the day. Thank you everyone.
Good morning.
Good morning.
Thank you for coming today. I really appreciate it. Now, I want to get a handle on some of the crops. Forgive me if I don't say it exactly right. Citrusdal?
Yes.
So I'm a Citrusdal, so I know there's citrus. I saw some citrus trees coming in today. And I know there's some wheat. What other crops do we have? What other crops are we farming here? Table grapes? And wine grapes? Or both table and wine, or just wine? Table and wine. Table, wine. Any maize? Not so much of a maize? No. What else? Is that pretty much it? What about potatoes? A little bit of potato? So are these the basic crops?
This is Citrusdal, but from our area we also have tomatoes.
Tomatoes? Okay.
Tomatoes.
Some people say tomatoes, some people say tomatoes. I'm from Canada, I say tomatoes.
Yeah.
Forgive me.
So Rick is referring to what you guys have on your farm. Like
Yes, I want to know what you grow.
Yes.
Have I covered the basics? I got them pretty much?
So wine grapes?
Yes, yes, grapes, table and wine. Yep, good. That just helps me as I go through the talk today, which crops I can focus on a little bit more on some of the research and some of the data I'm going to have, that I'm going to put up a little bit later. You know, if there wasn't anybody growing potatoes here, I'm not going to spend a lot of time showing you potato data, because you don't grow potatoes. But if I have some tomato data, and I do have some tomato data I'm going to show you today, I'm going to spend a bit of time showing you that now, because I know you grow tomatoes. So, that crazy looking animal in the far corner there is called a Canadian beaver. And I put that on there because that's my country's mascot, is the Canadian beaver. It's found right across the country everywhere. It's quite a nuisance to the farming. They build dams that plug up rivers and streams and interfere with irrigation. When I grew up on a farm. And I worked on a farm where the beavers would dam the rivers, and as a young kid we got to go up and blast the dams. We'd go out with dynamite and blast the dams. And when you're 20 or 19 years old playing around with dynamite in the field, exactly, it can be kind of fun and a little bit dangerous. I still have all my digits though. I didn't blast them off. What I want to do today is share crop nutrition from an American perspective, a North American perspective, in the hopes that we have some stuff that I can offer you that you might be able to take a little bit of knowledge, something that you haven't thought about before, back to the farm to do better. Our goal at AgroK is to help you do better. Because you're a customer, and if you're not doing good, we're not doing good. So we want to help you do better. First, let me say, AgroK is proud to be associated with EcoFit, Romina and her team. AgroK is a company based out of the United States of America in Minneapolis, Minnesota. All the products you're buying from Romina are made in Minnesota and then shipped over here to South Africa for you to use. And we stand behind our products. And we stand behind Romina. We're proud to have EcoFit representing us across South Africa. So thank you. AgroK is not a big corporate giant. It is a family-owned company. The Mayo family, Lohini and Chapman Mayo, own the company. And we have reps right across the US. I'm the Canadian rep and the international rep that gets to travel around and spend a lot of time in airplanes and seeing different crops from all over the place. We certainly have business in North America, so the United States and Canada, but I also do business in New Zealand, Australia, Ecuador, and of course, most importantly, South Africa, right? So we work in a multitude of crops. So I get to work in everything from citrus to table grapes to wine grapes to wheat and canola, right across to different tree fruits, from apples to blueberries and raspberries and strawberries. We work in a number of different crops right across the globe. That's where I come from. That's very close to my home. So I come from Western Canada in the province of British Columbia. So we border the Pacific Ocean, and we're in an area that is roughly 400 kilometers inland from the ocean. It's a drier area, not so dry as some areas around Uppington and around South Africa, but drier. We don't produce table grapes in that area, but we produce some really good red and white wines. So we have wines we have wine grapes in that area, and that's what you see there. That winery is called the Quailsgate Winery. They have production in British Columbia and also in Australia. And so we produce a lot of nice wines there. But we also have apples, pears, stone fruit, from peaches, nectarines, right across. And that is the Okanagan Lake. So that lake is roughly 200 kilometers long. It's a big lake. But that's home. And we get very warm in the summer. Typically in the summer months, we can see temperatures in around 29 to 35 degrees. But then we get colder in the winter. I find the weather now is quite comfortable. Our winters typically get down to about minus 15. And we do get the odd cold snap of minus 32. But then when I go into what I call the Canadian prairies, which is the center part of the country, where we grow most of our wheat and canola, my first trip as an agri-K rep to Saskatchewan, which is where most of those cereal grains are grown, when I arrived to meet my distributor, it was minus 45. I did not want to get out of my car. You go in for lunch and you leave the motor running so the car is still warm when you come back out. Kind of thing. So 15 to 16 degrees, I don't know if I put my shorts on. Except I'm coming into winter, so I've just finished my winter, so my legs are quite white. I need some sun. Okay, here's what I want to cover today. I'm looking for questions throughout. So if you have a question for me, don't wait till the end. Interrupt me, put your hand up, get my attention. And ask your question right away. I find that's the best way rather than waiting till the end. So if you have questions, bring them forward. What I want to talk about today, and what I'm going to cover today, I'm going to talk about what I call the 5 R's of plant nutrition. And I'm going to explain what those 5 R's are. And I'm hopefully going to do a good enough job explaining them that you'll understand why they're important and how we apply them to crop nutrition and fertilizer on the farm. I'm also going to show you some research to back up these principles. I want to show you how these principles are making a difference on the farm in the research that we have. And then I'm going to spend a fair bit of time as well talking about NovaCrop SAP analysis and how we're using that SAP analysis to drive the 5 R's, to help us do a better job applying those principles of the 5 R's. Because the better we can apply those principles, the better you're going to do at the farm. So I'm not going to spend a great deal of time. This is not a product talk. This is a nutritional talk. I will mention some products, but I'm sure you guys hear a lot about different products and sales reps talking about products. So I'm not going to spend a lot of time doing that today. You get enough of that. What I want to talk about today is nutrition and how to do a better job with the principles of nutrition. So when I do mention a few products, I'm not going to go into a lot of detail. If there is something that you want more information on, ask. I'm gladly going to share, but I'm intentionally focusing on nutrition.
While Eric goes to the next slide, if anyone feels like uncomfortable to ask the question in English, please ask in Afrikaans, I'll translate, sir.
Yes, please, yes. The only silly question today is the one not asked, right? So here are the 5 R's of plant nutrition from an agro-K perspective. We're talking about the right nutrient at the right time, in the right form, the right place, and the right mix. I consider these fundamental principles. Whenever I'm working on a farm and doing plant nutrition, and I'm working on your fertilizer, these have to be at the top of the mind. So if you have a crop advisor, an advisor you're working with that puts together your fertilizer program, top of mind should be these 5 principles. And when you look at that program that crop advisor gives you, if you're thinking about this and the program doesn't make sense, ask yourself why and try to understand it better, because it needs to apply to these. The better we apply these 5 principles, the better you're going to use your fertilizer, and the more efficient your fertilizer is going to be. And with the cost of fertilizer today being what it is, we can't afford to waste it anymore. We can't afford to pour it on anymore when we don't need it. We need to be more precise with our fertilizer. Those days of luxury are over, at least for the next while anyways, next number of years. We need to be more precise. This is how we can be more precise, by applying and understanding these principles and then applying them. So, right nutrient at the right time. We're trying to make sure that the plant is being fed what it needs when it needs it. When I was back on the farm growing up, Dad would just apply the fertilizer in the springtime because it was spring. And you just apply all of it in one go. Well, those days are over. Like I said, you're kind of missing those windows of opportunity. We have to focus to what we're trying to do, and we have to keep that in mind. So how do we do that? Well, we need to understand the different growth stages that the crops we have, whether it's citrus, whether it's wheat, whether it's potatoes or tomatoes, what stages are we going through? We need to be aware of that. We often talk about growth stages when we talk about an insect or a disease. Yeah, at this growth stage, I'm going to get this disease. At this growth stage, I'm going to get that insect pressure, which is great. We now need to change our thinking. We need the same thinking has to go into our fertility, into how we apply our fertilizers. Putting it all at the same time anymore is not efficient. It's too costly. We're not maximizing our yields. So we need to be thinking, what growth stage am I at? And what do I need to do to support that growth stage? And then we have to think about, okay, what role does each nutrient play? You know, we used to focus on the big 3, N, P, and K, nitrogen, phosphorus, and potash, right? Well, what about those micronutrients? Quite often, it's the micronutrient that helps the plant use the macronutrient. A good example of that is nitrogen. Nitrogen is very important, right? It's a fundamental building block to every part of that plant that you're growing and asking to produce a crop. So it's pretty critical. Without molybdenum, without moly, that nitrogen can't be metabolized properly. And you'll put more and more nitrogen on, thinking I need to put more on, I'm not meeting the needs. When in actual fact, you've got way too much nitrogen, but you're missing the moly, the molybdenum. And do you know a lot of soil labs, now if you're using I know some people will use a North American lab. I think Brookside is where some people, it's a good lab. I'm not sure about the labs here in South Africa. The UK, the Netherlands test, but not a lot of labs test for molybdenum. And you know that moly is one of the essential nutrients? And it's key to nitrogen metabolism. So if your soil test doesn't have molybdenum on it, you can ask the lab to do that. And it's not an expensive test. The labs that I've asked, why aren't you testing for molybdenum? The answer I get from the lab, because the farmer's not asking. It's not an expensive test, but it's critical for how nitrogen is utilized. So these micronutrients have a huge role to play in how the plant can use the macronutrients. So we need to understand that and tie that into the growth stages. So coming into bloom, when we want to make sure we got a healthy flower and good healthy pollen, viable pollen to maximize my fruit set, I want to make sure I got enough boron. It's kind of important. Boron has a huge role on pollen viability and fruit set. We want to be thinking about these things. So, here's an example of the right nutrient at the right time. Calcium. In North America right now, there's a big wave. Everybody's talking about calcium, calcium, calcium. We need to do our calcium. And by the way, if you have storability issues, if your fruit isn't making it to the grocery shelf, wherever you're shipping it to, whether it's down the street or up to the UK, where it has to go in the grocery store after sitting on a boat much longer than usual now, right? Everything's shipping. I mean, I ship fertilizer here from North America. It takes twice as long now as it used to. Well, the fruit, I'm sure, going north takes twice as long as well. Storability, calcium is part of the key to that. But when do you apply it? When do you put on calcium to help with storability? If we want to make firmer fruit, whether it's a table grape, whether it's citrus, whether it's potato, we gotta put it in during that cell division. Cell division, depending on the crop you're working in, happens within 3 to 6 weeks after fruit set. That's your window where you want to make sure the plant is going to get as much calcium as possible. Now, during that window of opportunity, when we're trying to get the calcium in, we want to do foliar sprays. Foliar calcium going in will help, because calcium is not mobile. It only comes in with water. And that water pump for the plant, whether it's a citrus, whether it's a potato or a tomato, is the water leaves the leaves, so you got transpiration occurring, respiration, transpiration occurring, the leaves breathe. Water is expelled from the leaf, and that sucks more water into the roots, and that's where the calcium comes from. And then we foliar to take advantage of that window of opportunity. So we do got to make sure we have a rich soil environment for calcium. So soil health is part of this. We got to make sure we have the balance in the soil. Then we got to make sure that the nutrients getting in to help. But for calcium, this is the window, fruit cell division window. After 4 to 6 weeks, now we're doing fruit bulking. So as that grape gets bigger, from that little green BB stage, as it gets bigger, that's not cell division. That's bulking. The time to get calcium into the fruit is over. You've missed the window if you haven't put calcium in until that point. So calcium goes on much earlier than normal. On a lot of crops, wine grapes and table grapes would be included in that. We start ramping up the calcium as we're coming into bloom, so that there's lots of calcium around in the plant as we're entering into bloom and moving through bloom. So we have it there in luxury, so that it's there as the cells are being formed and it can get into that cell wall structure. So it's all about that timing. Now, we do put on what we call maintenance calcium sprays after that fruit cell division window is over. But that calcium is no longer going to the fruit. See, what do we have happen after fruit cell division? After we've got that fruit cell division, we get some rapid growth, right? We get more growth, more leaves, more tissue. Well, that's all cell structure too. Those cells also need calcium. So we're putting on calcium at that time to meet the needs of the growth. Because if we don't, that pump becomes a negative pump, and it can actually pull the calcium out of the fruit. And we don't want that. We want to keep that calcium in the fruit. This is also an opportunity as we go through that bulking stage where we need to make sure that we're managing our insect pressure. There's been some recent work done that's really fascinating now on, do you know that when an insect bites a leaf, the first thing the plant does within minutes of that aphid biting the leaf or whatever it is the plant sends calcium to heal the wound. So that immobile nutrient that sits in the plant that we have trouble getting in, as soon as that plant is attacked by an insect, it starts moving the calcium out of the fruit to the wound. Immediately, within a few minutes, they've actually tracked it with radioisotopes, and they've got it on video now, you can watch it, and it's just fascinating. The insect bites, whether it's a caterpillar or an aphid, and within minutes, it lights up and it's moving the calcium to get to the wound to heal the plant. So we have to put those maintenance calcium applications on to make sure that the calcium that we work so hard to get into the fruit stays into the fruit. Now, Right form, right place. What do I mean by that? Well, in a very simple message, are the fertilizers that you're buying, those expensive fertilizers that you're buying, are they getting into the plant? Because if they're not getting in, they're not working. The fertilizer, the nutrient you buy, has to get in to do the job. So is it getting in? Form of the nutrient you're applying matters. And a good example is chelates. The most common chelates are EDTA and DTPA. So if you're buying an iron EDTA chelate and foliar applying it, I'll be blunt, you're wasting your money. There's a bunch of research, you can Google it online, that all speaks to the fact that iron chelate and zinc chelate, EDTA, do not penetrate the leaf. Now, does that mean that they're bad products? Heck no. Iron EDTA chelate is a good product. But what was the chelate invented for? To keep the nutrient stable in the soil so that it wouldn't get bound up in the soil and remain plant available, right? It was never meant to go on a leaf. But it's got a high percentage of nutrient. We can dissolve it in water and spray it on the leaf. But there's good research out there that shows it's not getting into the leaf. It's a good product, but it's meant to go through your drip, the fertigation, to the soil. And that, it does a very good job of. And regardless of your pH, whether you have a fairly high pH where the iron is going to bind up or not, it keeps it stable and plant available and the roots can take it in. So there's an example of using the right form in the right way. It's not that it's a bad fertilizer, but we got to use the fertilizer in the way it was meant to be used. And we got to be understanding when we buy a product, how it's meant to be applied. Here's some research that AgroK does. One of the reasons I work at AgroK, and I've been with AgroK now for 7 years. I've been doing crop nutrition for 32 years in North America primarily, but 32 years of crop nutrition, the last 7 with AgroK. One of the things I'm proud of with AgroK, we take 5% of our earnings every year and invest them back into crop research so that we can show you, the farmer, how to do a better job. We invest in your industry because we want you to do better. And here's a research project that we did on blueberries, but the principle is still the same. Now, forgive me in that in North America, we do things backwards. We don't represent phosphorus and potash as straight phosphorus. We recognize it as the oxidized form. So phosphorus to us is P2O5, not true P. So you guys are a little bit ahead of us in North America on that front. So this is looking at it as P2O5, but in this case, we have 2 different forms of phosphorus. The 105210 is what we call a polyphosphate. The 9243 is an orthophosphate. Now, a polyphosphate is a chain of orthophosphates. And this trial started because I was working with a researcher, and she told me that you cannot foliar apply phosphorus. It doesn't get in, it doesn't work. And I said, yes, it does. She said, no, it doesn't. I said, yeah, it does. You just got to use the right form. Form doesn't matter. You can't get it in. Yes, you can. As a matter of fact, I'm going to hire you as a researcher to do a research trial so that you can prove it to yourself. So we actually hired her to do a research trial, and she set up randomized blocks. She did it proper, randomized blocks, blind trial, and we put on the same amount of phosphorus. So we put on 2.6 pounds of actual oxidized phosphorus, forgive me, applied foliar. And then we went back in at 3 days and 10 days post-application to compare the difference between the treated blocks and the untreated control, right? The untreated check. Here's 3 days after the application. Now the way to read this is the green bars, and we looked at young leaf and old leaf separately. And the reason we did that is phosphorus, as much as it's not overly mobile in the ground, it's actually very mobile in the plant. So if you have excessive phosphorus, it's going to build up in the old leaves and then move up to the new growth as it's needed. So we looked at it both. So there's the 9243, the orthophosphate in both cases, and there's the 105210. Big difference in 3 days. So 3 days after application, the young leaf is 114% more than the untreated block, I should say, at 37, and 101% to 56% in the old leaf. Which one moved the needle? Which one did a better job? Look at it at 10 days. Look at the difference. So now at 10 days out, we're at 84% more phosphorus in the young leaf over the check versus 9% of the other and 38. So at the end of the day, my message is this: the percent nutrient matters. So you can look at the percent on the bag, it matters. But the form matters more. A lot of our aggregate products are not high analysis products. A lot of them that you'll see on our product lineup are simple sugars. We have two technologies. We've got the DL Vigors that are simple sugars, and the simple sugars are dextrose and lactose. So we're able to create a mineral nutrient and combine it into a complex of simple sugars with almost 100% uptake by the leaf within the first 48 to 72 hours. It's in. And then the other one we have, which we're going to talk about in a little bit, is the calcium phosphite. And that phosphite is really neat to play with, particularly in potatoes. It can make a huge difference. But the key messaging is the form of the nutrient and the way you use it is more important than the analysis on the bag. You can get a 36% product or a 30% product, but if it's not getting in, does it really matter that it had a high analysis? You can buy zinc oxide out there. There's lots of zinc oxide type products or magnesium oxide products on the market. They don't get in. That's why they're cheap. They're high analysis and they're cheap, but they don't get in. So we need to be aware of what we're applying. And then the right place. Now, kind of use the seaweed here because I wanted to talk a little bit about seaweed, but it underlines the score of how where we put what we're applying matters as much as what we're applying. So let's take seaweed. We have a product called Symspray, and it's oscophyllum nodosum, so a little bit different than Kelp, but still kelp, high in gibberellins. And when we apply that seaweed as a foliar to the leaf, the impact on the plant is a release of stress. So we're managing stress. So if you're having some hot weather and you're under some drought conditions and you have a higher stress environment for the plant, we foliar apply the seaweed and we get stress reduction. Now, if I take that same product and put it through the drip to the roots, I'm helping improve root function, which in a side effect helps with stress, right? Because a better root function is going to help with stress, but it's really focused on helping with root function. So where I take the product and how I apply it has an impact to the outcome. Zinc is another example. If I fertigate zinc, like a zinc chelate EDTA, which is good for the soil, right? If I take that zinc chelate and fertigate it, I'm putting it in the root zone. I'm going to help grow root tips. That root, as it grows out, that root tip, zinc is what helps that root tip push out and grow. Why is that important in storability and fruit quality? Because most of the calcium is coming in through the roots. The foliar is just the augment. And where does the calcium come in through the roots? The root tip. So when we talk about getting calcium in, we need the root tip to get the calcium in. If we're deficient in calcium in the root zone, we're not growing more root tip, we can't get the calcium in. It can be around the roots, but if it's not at the root tip, then the root tip kind of closes off, it can't come in. So we fertigate with zinc when we're pushing roots to make sure we're getting new root tips that can absorb the calcium during that fruit cell division window. And then if I take the same zinc, but in a different form, like a simple sugar, and I foliar apply it, now I'm building vascular tissue. So when I say zinc foliar, think big leaves. And the bigger leaves mean we have a bigger factory for sugars. So if you want sugars in your grapes, you need big leaves, right? Where are the sugars coming from? That thing called photosynthesis, right? From chlorophyll? Well, that's all driven by the size of the factory. If we can get bigger leaves, by the way, will also help regulate the temperature in the canopy of the crop. So if you're in a hotter environment, things like South Africa, if I have a bigger leaf, the plant is going to help regulate the temperature within the crop better. You need early season zinc when those leaves are developing. So, when you're coming into that 15 to 20 centimeters of new growth in the springtime, you should be putting on zinc foliar zinc, because we want tissue, we want big leaves. So we need to apply it foliar. You put it in the roots, great, you're gonna help with the root growth, which is great for calcium, but you're not gonna get the sugars the way you need. So you need a time where you're putting foliar zinc on as well to get big leaves. So it matters not just what the nutrient is, where we're putting it to get the outcome we're looking for. Another thing with our seaweed, by the way, with the Sim spray, Ascophyllum nodosum is an ethylene inhibitor. It works great on grapes when you're trying to stretch that bunch out. And the secret to doing that is to do a low rate, don't need high rates. With seaweeds, never just put 2 or 3 high rates on. It's a waste of product and it's a waste of your money. Seaweeds work best when you do a low rate frequently. Kind of, I tell farmers, a couple glugs in every tank kind of idea. Just a little bit frequently from the beginning when you first start spraying, whether it's a pesticide or nutrient, just a couple glugs when you start, and keep doing that regularly up until you start seeing color. And why do we stop when we start seeing color? Because ethylene. We need that ethylene production to build the colors and get the transfer from sugars, acids to sugars, right? We need that transfer to happen. That requires the production of ethylene. If we keep doing seaweed at that point, it's not going to work. It's going to slow the whole thing down. In apple crops, we use seaweed to slow the crop down so we can plan our harvest. But grapes, when you're trying to get them to color and build the Brix, wine grapes, we need the ethylene production. So be aware, but use it to its advantage. Know that it's an ethylene inhibitor, but know that also, if you do it regularly in season, you'll stretch that bunch out, which improves air circulation, which helps with botrytis, and helps with uniform maturity of the bunch. So they don't have that little green ones in the middle of the nice colored ones. We can stretch that out, get better uniform light penetration, airflow, less disease pressure, and better color, better uniform color. Make sense? Right mix. This is a fun one. Because what I mean by this is some nutrients get along well together, some nutrients play well in that sandbox together, and some just don't. Some antagonize, they hurt each other, they don't get along at all. And by being aware of which ones work together and which ones do not work together, we can avoid doing things that aren't going to work, that are going to kind of cancel each other out, and do things that are going to help each other. Here's a big one. Actually, there's 2 big ones. These are the 2 big ones. If you take away nothing else today, if you find this new, this is big. The first one is potassium and calcium. Early season, when that fruit cell division is going on, potassium will prevent calcium from going into the fruit. So if you have a lot of potassium around, it will prevent the calcium from going into the fruit during that fruit cell division window. So if you're applying calcium or potassium foliar early season, you're actually not just doing nothing, you're actually hurting yourself. And you should not do that, simply put. Because that potassium will prevent the calcium from going into the fruit cell wall structure, and it will prevent you from getting good quality fruit. I've seen growers that would apply calcium during that fruit cell division window every 5 days. And they were putting it on with a quality potassium product, and then they have soft fruit issues at the end of the year. And they'd come in, Rick, you said if I put on my calcium every 5 to 7 days, I wouldn't have soft fruit. I've got soft fruit. Yeah, but you put it on with potassium every time you put it on. Oh. So early season potassium needs in the crop are actually very low. We don't want a lot of potassium around. And then here's the synergy, and this is a big synergy. Calcium and boron. You know, by putting a little bit of boron with your calcium, when the boron is needed, you're increasing the uptake of that calcium by 10 to 15%, just by having a little bit of boron. That's a pretty neat synergy, because at that early season stage when we're trying to ramp and get higher calcium in the plant coming into bloom, isn't that when we want boron around? So putting a little bit of boron, the right boron source in with your calcium is going to improve that uptake by 10 to 15%, and you need both anyways, so put them together. Now you got to be a little bit careful with that, because later when you start doing those calcium maintenance sprays, and the boron isn't really needed so much anymore, you can get into trouble. And boron toxicity, essentially you get cells die. You say cell mortality, the cells start dying, the leaves become quite wilted. I've seen boron levels so high where the leaves look like wet toilet paper. It just wilted right over. And when we send them to the lab for analysis, half the sample's rotten before it even gets to the lab because the cell structure's already broken down. So we need to avoid that boron toxicity, but having the right amount of boron is pretty important. In the early season, it helps with that cell structure, which is why there's a synergy with calcium. It helps with the pollen viability and the flower health. But boron also plays a role. During the day, the leaf, that factory called a leaf, is making sugars. At night, it moves those sugars and starts digesting it. That movement of sugars, boron plays a huge role in that movement of sugars. If you're deficient on boron, you're not going to move the night sugars, and you're going to get a buildup of sugars in those leaves over time because the plant can't feed on them, they can't move it. So we don't want to be deficient in boron, but we don't want too much boron either. And boron is a true micronutrient. Macronutrients, right? The big ones, macro, big, micro, small. Boron is a true micronutrient. We don't need a lot. It goes a long ways, but we do need it. So that would be a big one. Here's some research. Now, this is on potatoes. This was done in Idaho. Now, in the United States of America, Idaho is called the potato state. 80% of the French fries served at McDonald's right across the US come out of Idaho. This is the big state for potatoes. And the big guru in potatoes in Idaho State is this gentleman right here, Dr. Jeff Miller. Yeah, at this point he was working at the University of Idaho. And what I want the takeaway here is there's your calcium levels in ppm, there's your untreated check at 9.12 ppm. Phosphorol is a potassium phosphite. So it's a potassium. And remember I told you potassium hurts the movement of calcium. Well, look at what the potassium did here. It's got less calcium in that tuber than the untreated check by a fair little bit. Now we went on with a non-phosphite, or a phosphite that doesn't have potassium. System zinc is a zinc phosphite. And by the way, you're going to see some research in a minute where we did the same thing with calcium phosphite here in South Africa. Remind us of a trial here in South Africa using the calcium phosphite and was able to duplicate it because it wasn't potassium. But look when we got away from when we moved away from potassium, look at that, 9.52, 11.41. We increased the calcium in the tuber by 25%. Now potatoes, I know, spent a few moments talking about potatoes. Potatoes are a weird plant in that we cannot, and we tried, you cannot foliar apply calcium to a potato and get it to the tuber. The way the tuber forms, the calcium won't move to the tuber. You can spray calcium till the cows come home. It will not go into the tuber. I don't care how good the calcium product is. It will not move, even a phosphite calcium. And phosphites move in the plant. Phosphites move in what we call the xylem and the phloem. So phosphites will move with the water and with the sugars. So it moves up and down in the plant. So we have a calcium phosphite that we thought would move the needle nicely as far as putting it doesn't, that calcium from that product will not go to the tuber. But the neat thing about a phosphite, when you have the calcium phosphite to play with here in South Africa, is registered and available, is we get what's called a phosphite root flush. So when you put on a phosphite on any plant, it creates a root flush of new growth. New feeder roots that come out into the ground. And as long as we have good calcium in the ground, so we put on our gypsum or whatever to make sure we've got a good source of calcium in the root zone, we can go on and the potatoes right at hooking, so early tuber initiation at hooking, put on a phosphite that's non-potassium, create a root flush, and uptake calcium through the roots naturally. But it's that phosphite root flush that's doing it. So when we come out ahead by 25% more calcium in the tuber, that's how we're doing it. We're actually pushing the roots to grow more roots and letting those roots pull the calcium in to get it into the tuber. That's the only route, is through the roots. It won't go through the leaf. And that's how we're doing it. But that same root flush can help. If you're starting off your season on your wine grapes a little bit cold and you want to wake those roots up and get them growing, we put on an early season phosphite, calcium phosphite, to wake the roots up and get them going and functioning a little quicker. So if you got cooler soils and maybe they're damp and wet from the rainy season still, we can use the calcium phosphite to wake them up and get them going.
That is the monocalcium phosphate.
Sorry?
Is that ammonium, monocalcium phosphate?
There is a, yes, yes, it would, yes, yes,
Okay.
you're right, there's a little bit of ammonium phosphate in there.
Mono.
Oh.
Monocalcium phosphate.
Instead of dye, just mono.
No, this is a combination of mono and di phosphate.
Okay, both.
Yes,
Together with ammonium.
yes.
Okay.
It's still the sugar base, basically.
No, this isn't sugar base, this is phosphate base. This is a phosphate base. Yeah, this is a true phosphate base. Yes, it's the bigger Kelp is the main, but it's not a sugar base, it's a true phosphate complex. So there's no potassium in there at all. And what a lot of people will do, and you have to be careful, and you'll see it on the label, is potassium phosphate is the cheapest phosphate to make. It's the easiest to make. Getting a stable calcium phosphate as a complex is very difficult. So what a lot of people will do is they'll make potassium phosphate and take some non-potassium calcium and put it in the jug and call it calcium phosphate. But what it really is potassium phosphate with non-phosphate calcium added to the tank. Now remember what I said about potassium phosphate, potassium antagonizing calcium. So that's not helping any because you got one antagonizing the other. And then you've got the phosphate. And with the phosphate, when we add a nutrient to a phosphate and it's not part of the complex, the phosphate will piggyback it into the leaf, but it immediately drops it. Where if you want to move that nutrient around in the plant, it needs to be part of the complex. So when I say calcium phosphate, it's a true there's no potassium in there. And if they're using what I just said about potassium phosphate is the piggyback, you look on the label, you'll see potassium. If it's a calcium phosphate and it has potassium, that's usually what they're doing. They're using potassium phosphate because it's cheap, adding some non-phosphate calcium to the tank, and that's where you get the antagonism, and it's kind of self-defeating. Where when you look at our drug, there's no potassium listed because there's no potassium there. But it is ammonium and diammonium phosphate that's in there in the reaction, yes. That's the reaction. Absolutely. Good question. Any other questions? Okay, here's the trial in South Africa. You can kind of get a hint at how well the trial plot went. We did the exact same thing. We took the bigger CalPhos, which is your trade name for the calcium phosphate, and we applied it a couple times right at tuber initiation, so no potassium. And you can see the outcome. Look at the quality of the crop here. I won't spend a lot of time on this slide, but basically that green is clean fruit. That was the problem fruit. And the untreated block had a substantially higher level of unclean fruit. We got the clean fruit to 93.5% of the fruit were defect-free, the marketable fruit. And I think the control, what was it? It was 60% or less?
30% crazy.
Yeah, it was 30% less. So, and if you do want to see more of the data behind this, I brought my laptop, I'm happy to show you. And the trick to this was it was a non-potassium. So that's an antagonism we got to be aware of, is that phosphite antagonism. And then we went on later here, and we kept applying, we applied a little bit of maintenance calcium, and that was the simple sugar. So that's after we've had the tuber initiation and tuber growth. So during tuber bulking, which is the same thing on wine grapes with fruit bulking, we kept doing a little bit of calcium because we wanted to keep the calcium in the tuber that we got into the tuber through the roots. So we did a little bit of calcium to maintain that. We did a little bit of sim spray to alleviate the stress, the seaweed to alleviate the stress, and voila, you can see how it impacted big time.
So, are you going to talk about magnesium later? Or
yes, what would you like to know about?
Why did you have magnesium?
Good question. Leaf function. If we're bulking, we need the sugars. So we need to focus on those photosynthetic nutrients. Of course, the key photosynthetic nutrient is magnesium for chlorophyll, right? Center of the chlorophyll, magnesium with 4 nitrogen in very simplistic terms. So in this case, I focused on the magnesium to focus on that, but depending on your soils, we need maybe more than just magnesium to focus on. But with these soils, we focused just on the magnesium. Before we did the trial, I looked at the soils to determine what was low.
Typically in these sands, you have the wrong ratio between calcium and magnesium. There's lots of magnesium that's not necessarily usable.
Yeah. So it's the calcium that's important.
And typically bulking is from potassium, so maybe you have bulking.
Once we're coming near the end of fruit cell division, that's when we want to see the calcium coming into play. But we also need to focus on the leaf function at that point. So that's when we put on the magnesium, the iron, make sure the sulfur is there. All those photosynthetic nutrients that support the leaf making sugars, because it's those sugars that are going to give us the bulking, right? So it's all about that leaf function. We have micronutrient packages that do that. Zinc plus 5 would be an example of the new one that we're bringing in. So it's got elevated levels of zinc, but it's got the magnesium, it's got the manganese, it's got the iron, all to help with function and photosynthesis, right? You got iron, magnesium, and sulfur that are required for chlorophyll production. And then manganese is important for the photosynthesis to take place because you're turning water into sugar, glucose. And water is H2O, that H2O has to split. The hydrogen and oxygen have to come apart. The enzyme that does that is dependent on manganese. So if we're manganese deficient, that factory isn't working on photosynthesis, we're not producing enough sugars. And if we don't produce the sugars, we don't get the bulking in the color.
So that's more important when you're adding more potassium.
I would say certainly early in the process of bulking, yes, but as the process goes on, the demand of potassium will go up exponentially, and that's where doing the testing tells us. But yes, in the early stages of bulking, the leaf function is definitely more critical. And then as that bulking goes on, the potassium becomes more critical. In the early stages of bulking, just like the leaf transpires, so does the fruit. So there's transpiration of that fruit. So there's water leaving that fruit as it's small as a little green berry. But as that fruit expands and bulks, the transpiration dramatically slows down. And that's where we need potassium to push the sugars into the fruit. So initially, it's the water pulling it in, but eventually, we become more reliant on potassium to move the sugars into the fruit. And there's a shift as that fruit bulks. So there are the 5 R's. And if we apply them well, this is what we see. And I'd see this. In North America, this is what we see. And it's not that I've got special products that, oh, it's my products that did this. What has given us the higher yields? It's applying the 5 R's. Doing a better job applying those 5 R's. Right nutrient, right time, right form, right mix, right place. The better we can do those, we get higher yields. We get better quality. We get improved storability. I'm going to show you some data on blueberries where we improved the Brix. So we increase the sugars. And usually when you increase sugars, people think we get soft fruit. We increase the Brix, increase the sugars of the berries, and improve the storability at the same time. And it's not that, like I said, it's not that our products are special. They're good products. I'm biased. But it's applying these 5 R's. We need to apply these 5 R's better. We can't afford not to anymore. And then we get better quality fruit. We get healthier plants. Farmers that I've been working with for a while, where we're really getting the 5 R's right, will say to me, those trees, they just look healthier. I don't have as many problems. I have hop growers that I work with. I've got a 1,000-acre hop grower in the state of Oregon that I work with. And hops for beer, they apply a lot of nitrogen, over-apply nitrogen. And I had a hop grower where they had a block they gave me, we were doing SAP analysis, and I told them they have to dial back the N. Not as much N, not as much nitrogen. We need more of this and this. Half the block, they followed the recommendation. Half the block, they did what they usually did. This is what Dad and I have been doing for 20 years. Great. At harvest time, the block that them and Dad have been doing for 20 years, 5 miticide sprays, and the leaves were still bronzy. The block that they followed the recommendation, better quality cones, they had yet to cross the threshold for the first mite spray. And mite sprays in North America, each spray was about $53 an acre, times 5, versus none yet. Now, if we get this right, I'm not going to say you're going to have no disease or insect. That we talked about earlier. I don't believe that's reality. But the better we do this, the healthier the plants, the less disease and insect pressure we're going to have, the less desirable these plants are going to be for the insects to be there. They're not going to want to be there. They're going to move off. And here's the big one for this year that to me really resonates this year, right? Fertilizer efficiency. You can't waste fertilizer anymore. It's too expensive. It's like gold. crying out loud. You can't waste it anymore. So the days of guessing really need to come to an end. And so how do we do a better job applying these 5 R's of plant nutrition? So keep that question in mind. I'm going to answer in a minute. I want to show you some tests. Told you on the blueberries. So what's going on here, these are blueberries, and this is the percent difference over the untreated control. This work was done in the state of New York, which is a pretty big blueberry production area in the United States. And so what we're trying to do in this case is we were trying to improve the uniformity of the harvest so that the berries were ripe more at the same time. Kind of important, right? So we increased uniformity 237% by doing SAP analysis and giving them what they needed when they needed it, applying those 5 R's. So we didn't increase yield 200%, but we increased the uniformity when that crop was ready to pick by 200%. At the same time, we took Brix up 20%. And then we took storage post-harvest up 40%. Now this was done because it's doing lab work, right? So what they do in this case is they assess the berries going in as far as marketability going into storage. They put them in a cooler at 4 degrees Celsius for 2 weeks, take them out and measure the amount of berries that are no longer measurable or marketable. How much berries have degraded. And we increased that storability by 40%. And part of that is putting the calcium on without the potassium antagonism, with the boron, in the right window of opportunity. It's working with the 5 R's. That's what does this. Our products are good, but it's working with the 5 R's that produces results. So if you have storability issues, focus on those 5 R's. You'll get better storability. Your product will make it to market in better shape and last longer.
How many
It really depends on the crop. You don't need to spend on citrus. You know, on citrus, if we do 4 a year at key windows of opportunity during development, that would be plenty. I mean, the lab would like you to do a SAP analysis every 2 weeks, but I don't run a lab. We have no financial connection to NovaCrop, which I'm going to talk about in a minute. We're completely independent of them. We go to NovaCrop because they're the market leader. They've been doing SAP analysis the longest. And when we do blind trials to find out how the lab is doing, so what we do is we take a number of samples from the same tree and send them to the lab as different farms. Let's see how close they're going to be. Right? Right? That's what you should be doing, by the way. Test your labs, make sure they're accurate. If you're going to make decisions based on that science, you need to know you can trust it. How do you know you can trust it? Test the lab. Send them the same sample in different baggies and label it separately. See what they do. If they come back radically different, you may need to find a different lab. Now, in the world of lab science, if you're within 10%, 5 to 10%, that's pretty good. That's where you should be. So when you judge the lab, they said that was 100 ppm and now it's 110 ppm. That lab, no, that's within the margin of error. But if one is at 100 ppm and the other's at 200 ppm and they're from the same tree, time to start shopping for a new lab. So that's what we did with NovaCrop. And we still do that with NovaCrop. We send samples to them regularly, blind, to see how close they are. And we do the same thing. There's 5 different labs within the US that do SAP analysis. We won't work with any of them because they're inconsistent. They are that inconsistent from sample to sample.
So on that subject, how quickly can you send the lead to a lab? Are you going to talk a bit about that?
I'm going to talk a bit about that, yeah. I'm going to get to that, yeah. Because really, this is how we're using the science to apply the 5 R's. So that's kind of where I'm kind of leading into talking about the SAP, and we're talking about the sampling. On certain crops where we have the data, we do fruit sampling. I know there's some crops that we don't, but like even on potatoes, we're now sampling the calcium along with the specific gravities on the potatoes, because we want to know, how do you know if you've improved the calcium in the fruit if you don't measure it? If you're never measuring it in the fruit, how do you know you did better? Right now with citrus in Florida, the Florida citrus market is on its knees. Half the citrus groves are gone. And those that are left are broke because of citrus green. It's a huge problem over there. What we're finding out is a lot of it is nitrogen mismanagement. That's a big part of it. Now, is that the only part? No. There's more to it than that, but it's a big part of it. So fruit sampling. If your goal is to put more of something into the fruit, and if you're not measuring, even if you don't know if that measurement is high, low, or medium, you got to measure it so you have a benchmark, so you know when you're doing better. Tissue sampling. I think there's a lot of tissue sampling going on. I don't think there's a lot of petiole sampling in wine grapes here, is there? It's more leaf sampling in wine grapes? Okay. You're going to hear me talk in a minute. I'm dead set against petiole sampling. I'll tell you why I'm dead set against petiole sampling. I tend to be a bit opinionated, I'm sorry. I'm Canadian, I apologize a lot. Soil sampling. Please do regular soil sampling. I have some very good growers I work with. I have one very good grower, he's a pear grower, big pear grower, had problems in his Anjou pears. Trying to figure it out, so I say, go get your soil samples. Comes up with this nice binder. Everything was tabbed, everything was organized. Very impressive. His most recent soil sample in the field was 25 years old. Like, what? Minimum every 3 to 5 years. More intensive production, do it every 2 years. But at least every 3 to 5 years, get a new soil sample. And get a detailed soil, so you want all the micros. I don't know if the labs here give you an opportunity to save money by only getting a couple things on the test. That information is dangerous, because you're not getting the full picture. Here's an example of that. I have a beef rancher that I work with, and all what they did was get the simple test. So it didn't have everything. It had NPK, it had sulfur, it had organic matter and pH, but it didn't have anything else. And it was cheap, $30 instead of $90 for the full sample. I got called into the equation because they had 15 dead cattle that were full-term gestation, ready to calve out, and it was nitrate poisoning. And what had happened was, their forage, they were in a soil that's called a serpentine soil. Serpentine soils are like calcareous soils where the potassium or the calcium is really high. It's like 3,000 to 5,000 ppm, and the nitrogen or the magnesium is also at really, really high, and that's what a serpentine soil. So both of them are up in that 5,000 to 6,000 ppm range. Well, magnesium and potassium and calcium all have ratios together, and because he didn't look at the magnesium, he was just looking at the potassium. The 350, 400 ppm was great, and standing by itself, that was right. But because the magnesium and calcium were so elevated high together, that wasn't enough potassium in balance, and the plants couldn't metabolize the nitrogen. So the nitrogen was coming in and converting to nitrate and staying as nitrate, and we had nitrate toxicity in the cows that killed the cows, full-term calves. So you're losing a cow that's worth over $3,000 and a calf that has huge value as well, because they didn't spend an extra $50 in the soil sample. Get the detail. The smaller ones are going to get you into trouble, because you need those micronutrients to help the macronutrients get used. Those balances are important. And then the new one, I shouldn't call it new because it's not new, it's been around decades, but the added tool in the equation is the SAP analysis. So soil health and nutrition is important. We talked earlier before the meeting about soil health. It is critical to manage your soils. That microflora, those microbes in the ground, are what help the roots function and keep those roots healthy. And by the way, your soil, even if you're at 2% organic matter, do you know that 2 to 2.5% organic matter represents about 20 to 30 units of nitrogen on an annual basis? That adds up. That's free nitrogen. We manage the soil and manage that soil microbes. We can tap into that. How much phosphorus? And when we look at a soil test, it's measuring what is extractable nutrient that the lab technique was able to extract. That doesn't mean that's all the phosphorus that's there or all the potassium that's there. That's all that was extractable, that the test was able to measure. There's a whole bunch of nutrition that's there that's so tightly bound up with chemical bonds that it wasn't extractable to be measured in the sample. If we can improve that microbial health, we can release those nutrients and tap into them. Now there's one exception that I mentioned it earlier when we start working with soil microbial health and soil microbial populations, we can release phosphorus, we can release more nitrogen, we can release more potassium and more micronutrients. But the one nutrient that goes down with improved microbial activity is sulfur. The nutrients digest and utilize the sulfur, and it gets used and comes out of the ground. And that's food sulfur, so that's not elemental sulfur, that's oxidized sulfur, sulfate. So as we improve the soil microbial activity, we need to watch that, we monitor and make sure the crop is getting enough sulfate sulfur, because that sulfate sulfur is responsible for chlorophyll production and utilizing the nitrogen, right? That's why a sulfur deficiency looks very much like a nitrogen deficiency. So we got to manage the sulfur as the bacteria become more active. So that's something to watch for. So it was just a great way to measure nutrients, what's around for uptake. We want those balances. And don't ignore those base saturations on the test. When I was in college, I determined, and my dad always told me before college, you manage those base saturations, calcium, potassium, magnesium, and you manage those, everything else will look after itself. Yeah, right, Dad. I'm in college, I know better. It's the levels that matter, the base saturations don't matter. 32 years later, Dad was right, in a large extent. So we look at the soil levels, but we also need to look at those base saturations, because those are the macro cations, those are the macronutrients that are present in large quantities, and when they are in balance, particularly the calcium, when calcium is in balance for your soils, everything else starts to click better. The microbes are more active because they're feeding off the calcium. The microbes are more active, the zinc is more available, everything else clicks. Calcium is a critical nutrient in the soil to get into the right balance, and then the microbes do better, and everything else functions nicer. Now, what should that calcium be at? It depends somewhat on your pH. If you are at a pH of 5, what is pH? I'm going to start with pH. It's a measure of H positive versus OH negative, right? So if you have a pH of 5, that means you got a lot of hydrogen around. So you're never going to get your calcium levels up to 65 or 75% of the base saturation if you're at a pH of 5. Good luck, won't happen. But if your pH is around 5, you should be shooting for 35 to 40% calcium. That should be your goal. Now, if your pH is above neutral, so your pH is closer to 7 or 8 or 8.1, well, at that point, you should be shooting for 65 to 75% minimum in calcium. And that base saturation of calcium in your soil test tells you whether you should be putting on gypsum or not. I'm a huge fan of gypsum. It's the most cost-effective, and I think it's relatively inexpensive compared to fertility. Am I right? Here, South Africa would be the same? Yeah, so it's a cheap form of calcium, and you're getting calcium, you're getting that sulfate that I was talking about earlier, right? Calcium sulfate, that's what gypsum is. So we're not impacting the pH. And gypsum, as it reacts in the ground and that calcium and sulfate comes in, it's breaking down those soils. So when you have those soils that become like concrete, gypsum will help. And don't put on just one big application a year. Take that application and split it out over 2 applications. Do you want it at the beginning of the grow season and one at the end of the grow season? Get the benefit year long. Okay. Nutrient levels, base saturations, organic matter, pH. Get them all, because soil health is critical. And this is why. Now, we do have a product called Symbex. And what Symbex does is it keeps the fungi, very specifically the mycorrhizae, and the bacteria, and very specifically the aerobic bacteria. We want to stimulate the aerobic bacteria. There's two types of bacteria we're working with in the ground. We've got the aerobic bacteria, which we want, and then we have what's called the anaerobic bacteria, which anaerobic, no air. Well, if we don't have a lot of air in the root zone, the roots aren't healthy. Pythium, Fusarium, right? We start getting root rot diseases. So we don't want a lot of anaerobic bacteria, we want the aerobic bacteria. So what Symbex does, and one of the key ingredients in Symbex that does this is cobalt. And what the Symbex does is it enhances the aerobic bacteria and specifically the mycorrhizae. Now, in your more acidic soils where you're closer to 5, you're going to be relying a lot more on those fungi, the mycorrhizae are way more active at 5. And then once you get up to 7, 6.5, 7, now it's bacteria that are way more active. But we want to look after both of those. Now, we can put on a carbon source. We were talking about earlier sugar. It's a great carbon source to get your bacteria going, right? You feed sugars to the soil, molasses. Put it on molasses or humic or fulvic acid. Great idea. Problem is with those products over time, if you're not managing the air in the ground or managing that the aerobic bacteria do better, over time the anaerobic bacteria respond better to simple sugars than the aerobic bacteria. So the first year that you put it on, you're going to go, I have found the solution. Right? By year 3, it's not quite working the way it used to. That molasses must be cheap. Somebody's not selling me the right molasses. They're cutting it with water or something. By year 7, year 10, you've got problems. And I've seen it over and over and over again. And it's because the anaerobic bacteria do better on the simple sugars than the aerobic bacteria. The Symbex enhances the aerobic bacteria so that they do better than the anaerobics over time. And that's what you want. You want to do better on the anaerobics over time. Now that's more of an issue on a heavy soil and certainly a higher pH soil than a lower pH soil, but I mentioned it not knowing everybody's pHs. So you want to be careful on that. And certainly working the soil helps get oxygen as well. You don't necessarily use Symbex or any problem, but you want to be careful that you're not getting the soil to the point where it's sealing off and you're getting a lack of oxygen. However you do that, you can manage that. Like I got cranberry bogs that I work with on cranberries, which are very heavy peat soils. They actually use hydrogen peroxide. In the springtime, they inject hydrogen peroxide in the ground to get oxygen into the root zone. So however you do it, be aware that if you're using molasses or products like that, you got to make sure that the aerobic bacteria are keeping the cutting edge above the anaerobics. It's very important.
Ricky, is there a way that you can measure the bacteria in the soil of the soil sample?
Yes, and so I believe in what I call the KISS principle. Keep it simple. KIS. And there's a test that the labs can do, and it's called the Solvita test. Aerobic bacteria, Solvita, S-O-L-V-I-T-A, Solvita. Most labs can do it, maybe not all labs, but there are labs around that can do the Solvita test. And what it is, it's a measure of the soil, the aerobic bacteria breathing. They breathe aerobic, they breathe oxygen, just like you and I do. And they give off carbon dioxide. So what they do, you send the sample in, and some labs will do it as they do the detailed soil sample. They dry the sample down, they take a certain volume of soil into an enclosed chamber, wet it down, and then the microbes become active again. And they measure the amount of carbon dioxide that comes off that soil sample over a set period of time. That gives them an idea of how active the microbes are. So they're not spending as much money and going into much detail as identifying what you've got for microbes in the ground, but it gives you a general, quick and dirty picture. Do I got good activity in the ground or not? And it's relatively cheap.
So if you would want to do a sample and see how much good bacteria and bad bacteria in the soil sample.
The Solvita will tell you.
But that would be more expensive. You see.
No, the Solvita, it's cheap.
So this is,
It's the cheap one. The more expensive one would have them to do a complete assay where they'll identify the different bacteria present, good and bad. That's right, because it's microscope work. With the Solvita, it's an easy test. You can wet it down in an enclosed environment and measure carbon dioxide.
yeah, I'm farming organic farm for 7 years now, and this I think it's organic from 2010. I can't remember.
Okay?
And I want to do soil samples on the bacteria that he uses in the soil for
Right?
breaking up salts and stuff. You understand what I mean? I want to see if it's working.
Yes.
So, but that will be a different sample or taste.
Well, it's still going to tell you whether you got good activity or not. But
The key point
is that activity. Because if you got strong activity, it's helping. So the key point is, are the bacteria there and active? The Solvita will tell you that.
okay.
And it's a cheap way of doing it. Now the other way is, of course, what we'll get into next, which is measuring the sodium in the sap. Because if we're breaking it up and getting it out, it's not going to be in the plant so much. So that's another way of monitoring and how good it is my microbial activity in helping break down and flushing the sodium. Do I have a sodium problem in the sap or not? So those are two tools that are cost effective to look at.
Okay.
Fransha, in your from SAP analysis, I know in the stuff I sent through to yesterday, you can see a clear improvement from last year in some of yours, so you can specifically see those soft ions being reduced in the SAP. So that's a very good indication
Yeah,
of good work in the soil.
the product was better, yeah, I can say.
And to me, at the end of the day, the best indicator is what you just said. I have better product.
Yes.
And that's what I want to hear. And I'm not satisfied until I hear that. I've got the Michigan blueberry growers in the States, it's a blueberry cooperative. And 5 years ago, 6 years ago, they were having huge problems. The yields weren't there, the fruit was soft and mushy. Nobody wanted to buy it, they didn't have buyers because the fruit wouldn't hold on the shelf. It'd be rotten by the time it hit the store shelf. Grocery stores tend not to pay for rotten fruit. So we started doing SAP analysis work, we started getting things in balance. This year, the growers that were on the program, with the SAP analysis, their fruit was noticed on the packing line. So now the co-op that packs the fruit is saying next year, they will not take any fruit that isn't doing SAP analysis. It's getting noticed on the packet. I'd say with an apple grower, I work with a 1,000-hectare apple grower in Ontario and Canada. The packers can now notice the quality of the fruit coming across the line. That's what we need, right? It comes along with those, like, you talk about raisins getting a 90 to 92% pack out of quality. That's where we need to be. That's where the money is.
A quick question, maybe you can help with the, on my analysis, the organic nitrogen is every time is very high. So we tested more than a year ago, we tried with the passion fruit. I planted some, and the insect was impossible. We couldn't keep up with them. Organic, I can spray for the, there is some stuff you can spray for the insects, but not in that high level of activity. So, what I want to know is the natural nitrogen a good thing or not so good thing? My level is very high. I plant cover crops, clovers now.
Sure, which is good. Total nitrogen being very high is not a bad thing. It means you can stop applying nitrogen and let the crop use it, and that total nitrogen can build up over time. Like the plant slows down because of the weather. You're in a drought. The crop isn't growing as much. It's not even, the nitrogen can build up as organic nitrogen in the sap. It's the nitrate nitrogen that can cause a problem. And when that nitrate nitrogen gets excessive, that's where we get the insects.
Yeah.
So it's the form of nitrogen that's building up in the sap that we have to monitor and manage. And if we can get those nitrates down, the insects won't be the same issue. I won't say they won't completely go away, but the pressure will drop.
Measure ammonium nitrogen as well?
Yes.
Yes.
Yeah,
That's
so-
higher than nitrogen is good.
Ammonium doesn't cause the insect problem as much as the nitrates do.
You want one higher than the other.
Yeah, we like ammonium higher than nitrate. Absolutely. And in that drier weather, because you've got amino acids and proteins that are the building blocks of nitrogen, and those are the organic nitrogen sources in that sample. In a period of stress, that protein and amino acid will actually break down and release nitrogen back into ammonium form. So that ammonium doesn't just go up into proteins and amino acids, it goes the other way as well if the plant is under stress. So we can't get an ammonium buildup. And we gotta ask, why are we getting that ammonium buildup? Is there a cycling problem, or is it just the stress that we're gonna move through and get on with it, and then it will be fine? If it's slowed down due to stress, we can use nickel. And nickel, some countries, I don't know if South Africa recognizes it as essential. The US started recognizing it as an essential nutrient back in '86. Canada just calls it a heavy metal and they don't want it, which is stupid. Sorry, I can say that about my own country. But nickel helps with the metabolism of ammonium back into amino acids and proteins, which is where we want it. If it's in that organic form, it's not going to cause a problem. And then when the conditions become right for the plant to utilize it again, it's amazing how quickly it will drop. So I'm going to go into a bit of that, a bit more shortly. So,
Can I just ask a question? You're
Yes.
talking about the ammonium and nitrate
Yep.
ratios in the sap.
mhm.
But don't you guys look at the ratios in the soil as well? Do you measure nitrate N and ammonium N in the soil or not?
Tend not to, because it's so volatile, it's hard to get an accurate measurement from the lab. Even the total nitrogen from a soil sample, I don't ignore it, but I don't put as much weight to it, because how that sample was in that bucket a little bit longer and volatile, it can change so rapidly in the soil.
In more examples, you see the trend.
Yes.
Okay, in the soil, if you've got anaerobic conditions in the soil,
Yes.
you will not have nitrification, so you'll have a lot of ammonium in the soil.
Correct.
So that can also happen, that you then have a larger ammonium uptake than a nitrate uptake.
Correct. Absolutely. And your pH drives that to some degree as well, because the nitrification bacteria are more active over 5.5 and 6. At 5.5, you put down ammonium fertilizer or urea, it could take 10 weeks for it to convert to nitrate. You do that same in your soil, Wilfred, it's going to convert within 7 to 10 days to nitrate. So the nitrification bacteria are also pH dependent.
Absolutely, yeah.
And different crops, and this is where more work needs to be done about, because when you hear a fertilizer rep tell you nitrogen, walk away. They're wrong, because it's different forms, and different crops want different ratios of nitrate to ammonium to perform well. And we still don't know what those ratios are, quite frankly. Blueberries want 100% ammonium. Other crops want a balance, somewhere between that 50-50 to 70-30, depending on the crop, between ammonium and nitrate. You want both. Now, in a drier year, where it's drier, you want to focus more on the nitrates. In a wetter year, the ammonium tends to do better. And the reason for that is nitrate comes in with water, comes all the way up, right? So water comes in, it goes up, and then it pushes the sugars back down and around, right? Nitrate comes in with water up into the leaves, up into the new growth. That's where the nitrate gets metabolized, in the new growth. If you're putting on ammonium, it has to come up and then push down with the sugars, which takes more water. So in a drier year, we want to be heavier to the nitrates. Not all the way to the nitrates, we want the balance to some degree, but we want to lean heavier to the nitrates in a drier year. In a wetter year, if we can afford it, we want to lean more to the ammonium. And the reason for that is if nitrate is around, the plant's going to take it in. It's like me and chocolate. If you put dark chocolate in front of me, I'm eating it. I shouldn't, you can tell, I'm sorry, but if it's dark chocolate, I'm going to take it. It's like candy, you can't say no. Same thing with nitrates in a plant. If the nitrate is in the root zone, whether the plant needs it or not, it's going to take it in. And by the way, it's also going to bring in water with it. That's how we get soft fruit later in the season, is we start applying nitrogen later in the season, and it brings water with it, and we bulk those cells with water instead of sugars, and we end up with soft fruit. So we want to manage those nitrates to the early season, not late season. So calcium nitrate during the fruit bulking stage, soft fruit. And it's because of the nitrate. You think I'm putting on calcium and I get firm fruit, but you're also putting on a whole whack of nitrate that's going to come in a whole lot easier than the calcium because of the molecule size. So nitrates bring in water, and they're going to come in whether the plant needs it or not. Ammonium, on the other hand, is regulated. The plant will only bring it in as it needs it. And the nice thing about ammonium, NH4 positive, your soil, the organics in your soil, have a negative charge. So ammonium will leach less than a nitrate, because the negative and positive charge attract each other, right? Just like on a, using a magnet. So negative opposites attract. So nitrate will leach, ammonium won't leach. Do you have stabilized nitrogen products here in South Africa?
We do.
That's the principle to those stabilized nitrogen products. They have urease inhibitors to keep it as urea so that it doesn't volatilize, so you can irrigate it or get rain and move it into the ground within 10 to 14 days usually. And then they have nitrification inhibitors to keep it as ammonium for a period of time, and depending on how much inhibitor they put in there, anywhere from 6 weeks up to 13 weeks. And that's the principle behind that. It's keeping it as ammonium, which is a positive charge, so it doesn't leach.
It's going to be more expensive. Is the guy really going to live for it? It isn't very cheap.
And by and large, yes, you're right. And that's why when I'm talking ammonium, I'm not talking those stabilized nitrogen, I'm just referring ammonium sulfate. You still got the ammonium. It's not going to leach as readily as nitrate. Or potassium nitrate is going to leach way more than ammonium sulfate, because that the anemonical nitrogen won't leach as readily because of the positive charge. So, but it's the same principle behind that slow release.
You probably want to move on, but I would like to know if you would suggest that you use nitrification inhibitor to feed these crops that you're talking about now more ammonium than nitrate. If you use a nitrification inhibitor, that will be the result. But would you recommend that? Because then at the same time you will have less leaching.
In North America, we do, and yes, I do. It depends on the crop. Because you're putting it out. The whole concept is rather than doing 2 or 3 applications of a standard like ammonium sulfate, I can use a stabilized nitrogen, put on 1 application at a slightly higher rate and last as long. But that ammonium needs organic matter to go onto it, to hold onto it,
Yeah.
right? So if you're at 1% organic matter, that's not enough. So I used to sell a lot of stabilized nitrogen, Reflex and Umax. In the US, it's manufactured and provided through Simplat, specialty fertilizers. I used to sell a lot of it. And in my area, if I had 2.5 to 3% organic matter or more, it performed quite well and did exactly that. You could put on 1 app and make it last as long as 3. So yes, it would. But if you're under that, if you're at 1.5 or 1% organic matter, I wouldn't. Because there's not enough organic matter to hold onto that ammonium, it's still going to fall through.
Even if you've got clay soils?
No, it depends on, because if you got clay soils with some decent organic matter, so if you have clay soils we don't got good water movement through, that's a whole other issue, but you're right, it's not going to leach. So I'm concerned about having enough organic matter and that it's not going to leach. So if you have a clay soil where you're already having percolation issues, you're right, you're not going to leach it. But we want to help break down those clay soils, and gypsum will do that. That collotic soil, gypsum breaks that down over time, makes it easier for the roots as well. But while you're working with that, if you do have percolation issues with your water, you're right. If the water's not moving well, the fertilizer's not flushing away either. But then neither is your sodium. Right? It's kind of you're darned if you do, darned if you don't kind of thing.
Yeah.
Yeah. But the nitrification inhibitors certainly have a place in the market, as long as you're using it in the right application. So SAP analysis. What the heck is a SAP analysis? I like to say it's like a blood sample for plants. So what they're doing is they're taking a leaf sample and they're taking the sap out of the leaf and measuring the nutrient content of that sap to find out what's in it. That's what they're doing. And it has showing us different things. I've had it before I started doing SAP. I had 25 years as a crop advisor with doing nutritional work on farms, and I shouldn't say quite often, but more frequently than I care to admit, you look at the soil test and you look at the tissue test, and geez, it looks great. But why does the crop look so crummy? Right? Right? The sap is showing us why. Because just because it's in the ground doesn't mean it got in. And the tissue isn't giving us quite the right picture. And by the way, neither is the petiole. I'm going to go into that in a second. So the company that we have gravitated to, and I know Romina's using them as well, is NovaCrop. We have no financial connection to them whatsoever. I like how Friedhelm asked last night, he asked me, does AgroK have at least a gentleman's agreement with NovaCrop? No, we don't. The reason we go to NovaCrop for a SAP analysis is because they're the leader. They're the ones that have figured it out. We go to other labs and send them blind samples and they come back radically different. I do the same thing with NovaCrop and they come back the same every time. That's what we're looking for. We want to make sure that the lab is doing the work right. These guys have figured it out. I've yet to find another lab that can be that consistent over and over and over again. Now, they're very unique in that you can't go to another lab and use their targets. Because one of the neat things that NovaCrop has done is they've developed target ranges, optimum ranges for over 200 crops. By the way, including citrus, wheat, canola, potatoes, grapes. They've got targets for all of those. What are these optimum ranges? How did they come up with them? They're not just averages. Some labs, particularly some tissue sample labs, they'll take the average, right? Maybe they'll do 10,000 samples in a year. There's your average. What NovaCrop does when they're developing is they get those averages. So they take all the averages from all the citrus that they've sampled across the globe, and then they run them through a computer model that they've developed looking at key indicators. And a good example of it as an indicator, to give you an idea what I'm talking about, is when you look at young leaf and old leaf potassium, and they're almost identical, they're identical to each other, now you're in your optimum range for that crop for potassium. That's an example of one of the indicators. And they've developed different indicators and different ratios between certain nutrients that they're looking for that tell them you're in the optimum range. By running that computer model on those averages, they can get up to 80% accuracy in the optimum range. And then the last 10%, because they want to get into the 90 percentile range on accuracy for you, that last 10% is done in the lab. They do research. And they compare yields and quality of fruit and ongoing research. They just did an interesting research project that's just finished 3 years of research on strawberries, showing that applying less nitrogen and more key micronutrients actually improve yield. So nitrogen doesn't necessarily improve yield. It can be challenging. So they are doing the research to get into that 90% range. So that's how those optimums are developed. They're not just an average. They're averages that are then put through a modeling system on the computer to develop an optimum range that is then fine-tuned in research. And their extraction technique is unique, because when the sap comes out of the leaf, they're not rupturing cell wall structure very much, which is critical. A lot of labs rupture cell wall structure. And then they have a dilution process that is proprietary, that I can't tell you because I don't even know, that they dilute the sap to work with it, because sap is very sticky, and you can measure the nutrient content. And unless you know their dilution ratios, you can't use their targets, because their targets are specific for their dilution ratios and how they manage that sample in the lab. But the reason we're working with them is because they're the market leader, they've got the optimum ranges, they've done the work, and farmers that I work with on NovaCrop, what they tell me is, you know, Rick, you do a sample, you make a prescription, I apply the fertilizer, I look later, and you come back with the next sample, and you can tell me whether I did it or not, every time. Isn't that proof that it's working? Right? Now, with the sap samples, we're extracting it from the leaves, we're measuring the nutrient content. Now, there are some funny protocols when we do a sap sample. Those of you that have done them can appreciate. They need to be off within the first 3 to 3.5 hours of sunlight, between sunrise and 3 to 3.5 hours. And that has to do with leaf tension for the extraction. And also, what does that leaf do all day? It's a factory. It's working all day. So if you come at it at different times every time for your sample, your results are going to be like this. So part of the way we're getting accurate results is we're coming in the same window. In the morning, within the first 3 to 3.5 hours of light. That's when we're doing our samples. We're not sampling the petiole. We're only sampling the leaves, so we don't want petioles, no stems. And why is that? And this is where it comes to the petiole sampling. The petiole is a highway going into that sink called a leaf. Certain nutrients only travel that highway at certain times of the day. So depending on when you're taking that petiole sample, that nutrient may be traveling the highway or not. Well, to pick on boron again is a prime example. Boron only travels that highway in the morning and in the late afternoon. So if you're sampling late morning or early afternoon, you're going to show boron at incorrect low levels. The boron can be quite high, but because the boron is only traveling that highway at certain times of the day, you're going to miss it in the petiole. But you're going to catch it in the leaf sample. And that's why I'm not so good on the petiole samples, because different nutrients travel at different times of the day. It's a highway, it's not a sink. We want to measure the sink. Moving forward, tissue sample. What is a tissue sample? We're sampling the entire leaf, all the nutrient that's in the leaf and on the leaf. Roughly 80% of that tissue sample on a leaf is leaf dry matter. It's leaf tissue. All that leaf tissue has nutrient, but that nutrient is tied up in the tissue. It's no longer available for the crop. Only about 20% of that sample is sap. The rest is tissue. And by the way, it includes everything that's on the leaf. I used to have growers that were doing tissue samples, And the tissue sample showed great on the zinc. They were using a zinc oxide from one of my competitors, I won't name it, but they were using a zinc oxide, and the tissue samples looked high in zinc, looked amazing. Yet the fruit was deficient, like things were not going well. Well, the leaf sample is capturing what's on the leaf and in the leaf. Just because it's on the leaf doesn't mean it's going to get in, right? We talked about that earlier. So when we look at the tissue sample, I'd say we're looking backwards. We're looking a little bit backwards as that tissue was forming, because we're looking at 80% of the sample is tissue that's already formed. So we're kind of looking a little bit back to what happened over the last 4 to 6 weeks. With the SAP sample, we're kind of looking at today what's in the cupboard for tomorrow, because we're only looking at that 20% of SAP, that's it, and we're just focusing right on that, because that's what's available for the crop. That's what you have to work with, and that's what we're looking at. And then we do young leaf and old leaf separately to catch that nutrient mobility, so we can actually see if the deficiency is about to happen before you see it in the crop. Because once you see it in the crop, you're behind the 8 ball. Well, we can catch it now before. Here's a test. Let's talk about tomatoes. Or tomatoes. Tomatoes. This was done in Florida by one of my colleagues, Jeff Glass. And this was comparing a tissue and a sap. Now the interesting, and this was from the same plant. So he took all the leaves from the same tomato plants in a field production tomato planting, and just split them up between going into the sap sample and going into the tissue. Look at calcium. If you were going off the tissue sample on calcium, you'd say you were good. Well, most of that is caught up in tissue. He was good 4 to 6 weeks ago. And tomatoes, I'm sure you can appreciate, tomatoes blossom and rot. We all know that well. It's the same thing as bitter bitten apples, it's a calcium disorder. So calcium is kind of important. And this is, by the way, coming into bloom. So this is when calcium demand is going up. So 4 to 6 weeks prior, when that calcium demand wasn't there, his calcium was optimal. But as the demand goes up, he became deficient. There's your young and old leaf on calcium. Way down in the barrel, bottom of the barrel. Look at potassium. His potassium bottoming, but actually, in the young leaf, it's getting almost to the point of excessive. Remember what I said about potassium antagonizing calcium? So he's actually deficient, and his K is going to prevent most of that from going into the fruit. He's in for a whole lot of blossom end rot and a whole lot of hurt. Now, here's an interesting thing I haven't touched on yet. Manganese. Manganese is the regulator of potassium. What I mean by that, if the plant has low manganese, it's going to luxury feed on K. It will pick up way more K than it needs. If we can get manganese levels up, potassium is more regulated in its uptake. So manganese is the regulator of potassium. Now in your acid soils, if you've got a soil pH of 5.55, manganese is going to be readily available. It improves as you go below 5, but at 5 it's going to be pretty good. But if you're at 6.5 or 8.1 for crying, manganese is going to be available. You got to put it on. And if you put it in the soil, it's going to get tied up because of the pH. You got to fully apply it. And then your potassium will be in better regulation. So in this case, we had this farmer go in and apply some product. And what he did in this case, he put on the Vigor Cal Phos, there's your system, the calcium phosphite, to push the calcium up. And then he put on our phosphite manganese. Essentially, you put on a manganese, a foliar manganese. And let's see what happened. Calcium came up. Now, it came up in the tissue as well, but look how much higher that calcium came up in the sap. Now, that's calcium that's available for the tomatoes right now. And this same principle applies to your wine grapes, your table grapes, your raisins. This same principle is applying right across. Look what we did with the manganese. Shot it way up. Right? It was what we needed. It was deficient. Look, boom. One application. Boom. Actually, it was 3 applications. I lied. My apologies. 3 applications of Vigor Cal Phos at 1.24 liters per hectare. Same rate, 3 weeks of the manganese. But we shot it way up, and look what happened to the K. That's the principle of managing fruit firmness right there, everyone. That's a key principle. Manage your manganese, manage your potassium, and focus on your calcium, and you're going to end up way better. Focus on your calcium without managing your potassium and manganese, you could be doing a whole lot of money for a whole lot of nothing. This principle, and the SAP shows us this better than anything else. We've been talking about these nutrient interactions for years. We've known about them for years. We can actually see them in the sap. So, this is kind of long. I had a petiole slide in here, and that's my problem with petiole. Is key nutrients are only traveling in the petiole at certain times of the day. So it's hard to manage. So with NovaCrop, the reason we're with NovaCrop is they're giving us precise, accurate results every time. They have some sampling procedures for sampling, and we're doing old and young leaf separately. For your specific crop, Ramina has all the protocols. So we've got a nice little booklet for each crop, for wheat, for corn, for grapes. We have little booklets for you that you can have on how to sample each crop properly. How do you read a SAP report? Well, when you look at a NovaCrop SAP analysis, the first thing to know about SAP is that the plant is always trying to regulate the new growth at the expense of everything else. It's always trying to regulate that new growth. So if it's an immobile nutrient that can't come up from the lower leaves or other sinks, the deficiency is going to be right away in the new growth if there's a deficiency. If we've got a more mobile nutrient that can move, it's going to pull it out of those old leaves as a sink into the young growth as it needs it. It will pull it out of the fruit for crying out loud if it needs it. That's just a sink for it. It's always trying to regulate the new growth. And the other thing with the sap that's different than the tissue is the level of optimum range for a crop is consistent through the season. It doesn't change. And I know that's different, isn't it? Very different. Here's what does change, is the amount of nutrient to reach that optimum level. The crop is always going for that same optimum level regardless of the time of the year. But based on the plant demand, early season, it may take very little potassium to get into that optimum range. But later during bulking, it could take a lot of potassium to get to that same optimum range. So the target never changes, but the amount of nutrient to maintain that target or even reach that target changes dramatically through the different plants' physiological growth stages. And that's also where we see the differences in variety. So the varietal differences are also expressed in that way. So we're still going, if we're talking wine grape, we're still going for the same target regardless of the variety, but one variety may get to that target very easy for that nutrient, and another one could take a lot of work and always struggle to get there. So that's where we see the varietal differences, and that's something very unique with the sap versus a tissue. But it does allow us to go and do sap samples all year long, right? Once we have these to work with. And this is why we look at mobile versus immobile nutrients. When I started after college, this chart was nutrient identification chart. So you could put nutrient identification at the top, because when you're out in the field looking and trying to figure out what a deficiency was, if it was chlorotic at the top, it's either iron and sulfur. If it was down lower, it could be magnesium, right? Depending on where it was. Well, why? It's that principle of the plant's always trying to regulate the new growth and whether a nutrient was mobile or not. That's what's making the symptoms show up down here, because if it needs K up here, K is mobile, it's going to go up right up. Boron can't, so you're going to see it right away up here. So it's that principle of mobility that drives where we see those deficiencies, or toxicities for that matter. And then when we look at a SAP result, from the NovaCrop perspective, the middle column is your optimum range, then you got a low and excessive. Here's an example: light green is the young leaf, dark green is the old leaf. You can see in potassium, we're fairly strong, we're almost beyond optimum, but we still have some good reserves here. Look at potassium, look at calcium. Look at the big difference between the young leaf and old leaf. 459 ppm and 1287. Calcium moves with water, right? That's how it moves in. If it's all going in the young leaf and it's not going into the old leaf and not in the young, see that huge difference between old and young leaf? You know what that tells me? There's not enough water moving through the plant. Now, not enough water can be one of a couple things. Doesn't mean turn on your irrigation, because it could be Fusarium, could be Pythium. So you could have root rot issues that are preventing those roots from functioning. All I can tell you is when that calcium between old and young leaf get that big, there's not enough water moving through the plant. Now we've got to figure out why. Is it a root health issue? Is it an irrigation issue? Is it both? But the SAP can tell us whether we got a water issue or not by looking at the SAP, which I think is pretty cool. When we look at a SAP, these are the balances we're playing with. So the way to read this is, there's all your cations, your positive charges, right? The big ones. Calcium, potassium, magnesium, ammonium, NH4, right? monocle nitrate, NH4, that's a positive charge. All these are balancing and tug of war with each other, pulling back and forth with each other. Same thing on the negatives. Nitrate, NO3, sulfur, even chloride and phosphorus. By the way, chloride in the right amount isn't a bad thing. Chlorides give your fruit flavor. Thank you for coming. All the best. Sorry if I'm going a bit long, everyone. How am I doing on time? Do I need to wrap up in the next 10 or 15? Okay, I'm going to be done in 20. But chloride helps develop flavonoids. You want good tasting grapes? Don't be afraid to put a little bit of chloride on it. But just like our body, too much is not good. The right amount is good. How do we know? Well, the SAP will tell you. Because there's targets for chlorides in grapes. And what happens if we overdo those targets? Soft fruit. So calcium chloride is good in moderation. It helps with the movement. Chlorides perform a lot like potassium. It helps with the movement of sugars. It helps with the opening and closing of the stomata on the other side of the leaf for respiration and transpiration. And it helps build the flavonoids that give us the flavor that we need, which is good. But too much creates soft fruit. Fruit that breaks down faster in storage. So it may look firm going in, but it breaks down faster in storage. So we want to measure those chlorides. And on the nitrates, if you have a lot of nitrates, because say your pH is quite high, so it's converting quickly, and if you have a high organic matter, the antidote for high nitrates, by the way, sulfur. Sulfur. Put on more sulfates. There's no such thing as a sulfur toxicity. Yeah, you can go beyond the range that the crop needs, but even excessive sulfur that's 3 to 5 times beyond the range the crop needs is not going to be detrimental to the crop. And if you have a nitrate problem, getting those sulfur levels up will help push the nitrates down in the plant. So these compete with each other, these compete with each other. This group of micros here in the red, they compete with each other. There's a lot of fungicides, by the way, that have zinc and manganese in them. Manzate, Mancozeb, do you have those products here? Bravo ZN? Yeah, so a lot of fungicides, a lot of them have zinc and manganese in them. So what will happen is we'll get really elevated levels of zinc and manganese, even copper products, Quava, copper oxychloride, will get really high copper because there's nutrient in those fungicides. That's how they work. It's part of the active. And that's not bad, but that often puts pressure on things like iron. Even in acidic soils, I've seen copper and manganese so high because of fungicide treatments, you can't get the iron in. So these compete with each other. And then we have this group over here, iodine, molybdenum, boron, silica. They are the friendly ones. They don't really compete with anybody. There's some synergies, but they don't compete. So there's some synergies like calcium and boron over here that work well together. Calcium and silica is also a synergy. Silica, is there calcium silica here in the market or no? Are there any silica products in the market here that people are playing with? Okay, silica. We need to differentiate between organic silica and inorganic silica. Silica is a naturally occurring mineral in the Earth's crust right across the globe. It's very abundant. I think it's either the 4th or 5th most abundant mineral in the Earth's crust. But it needs to convert to an organic acid before the plant can take it in. That conversion is through microbial activity. The soil microbes feed on the silica, convert it to silicum, which is an organic acid, and it's in that organic form that it can come into the crop. That's how the crop gets the silica. Silica gives rigidity to that cell wall structure. Now, if it's an organic silica coming in naturally in the sap, which measures silica, by the way, we can have really high silica levels and still not an impact on the crop. Now, sometimes that silica is coming in because we've got a really active microbial activity, we've got a really healthy soil. So silica can be a measure of healthy soil. Yes, but the other side of that is if the crop is under stress, the plant's stress mechanism is also to take in more silica. It releases more hydrogen from the roots, which drops the pH and brings in silica as an exchange. So stressed plants will also breed high in silica. But when we look at the sap, we also have this thing called aluminum. Aluminum and silica high together tells me the crop's under stress. Silica high and aluminum low tells me the crop is just, the roots are functioning healthy and the microbes are doing well. So the aluminum goes high with the stress. And the reason for that is hydrogen extraction. The roots exude hydrogen under stress. And that brings in more aluminum. So if I see both high aluminum and silica, it's a stress reaction. It's a bad thing. If I only have high silica, it's a good thing. It tells us we got good root function. But now we take those inorganic silica, potassium silica, and when you apply potassium silica, the leaves get more brittle, right? They get scratchy almost on crops. You can see the texture change on the leaf because inorganic silica, the mineral silica that we foliar apply may not come into the roots very well, but it does penetrate the leaf very well. And you're losing cell elasticity. You're getting cell rigidity, but the cells can no longer stretch. So what we start seeing with too much of that is we start seeing fruit cracking and fruit quality issues and weird things because the cells can't stretch properly. So a little bit of potassium silica is a good thing. A lot of potassium silica is a bad thing. We're still doing research to figure out where that line is for different crops. On grapes, we don't quite know yet. On blueberries, we're finding it's about 5 to 6 applications. And we're finding that the ratio between calcium and silica is a 1 to 5, 2 to 5. So 1 silica to 5 parts calcium, or 2 parts silica to 5 parts calcium. Seems like the sweet, good ratio to be in. If you're beyond that on the silica to calcium, be very careful. You're going down a road that could lead to fruit quality issues and cracking specifically. And again, it's just, I had a blueberry grower that put on 12 applications of silica over a year. His blueberries were like marbles. They were rock hard at harvest time. Yep. Yes. I know what that means, I think. And then they put them in the hydro cooler to cool them off. And it was like watching popcorn. They all split. Thousands of pounds of blueberries all split. And that was because they lost that elasticity. So we want to be careful with the foliar applications. And by the way, if we push calcium too high with the inorganic silica, or silica too high with the inorganic silica, it starts hindering the uptake of calcium. So when we start putting calcium silica on, the silica will improve the uptake of calcium. But when we go too far with it, it inhibits the uptake of calcium. So it starts hurting you. So silica is a good thing, just don't overdo it. Where I like to see a lot of silica is during the fruit bulking period. Stay away from it during the cell division window, but during the fruit bulking period, there still is a little bit of silica being absorbed into that skin and having an impact and having a positive. But during the fruit cell division window, I tend to stay away from it, and what I found works best. Find for yourself. Big picture. So when we're looking at the big picture, the first 3 at the top of the big picture, the interesting one that I want to really focus in here are the total sugars. Measure of total sugars. Now, talking about John Kelp, if I can say for a minute, he's starting to say now more recently that Brix is a good measure for insect and disease pressure. As a matter of fact, there's even some ratio out there saying if your Brix is between this number and this number, you're low, and if it gets to this number, you're medium, or this number is high. And in principle, it's the right way, but wrong unit of measure. Wrong unit of measure, because what is Brix? Brix, soluble solids in the sap. Soluble solids, right? Well, what makes up your soluble solids? Sugars. NEC, your salts. So if it's high in the sugars, he's right. Less insects. If it's high in the salts, may not be right. Bingo. So what I like about the SAP is we got both of them. We can make that determination. If it's high salts, I'm not too worried about it, depending on what's causing that. And we can look further in the test to find out where what's driving that. If it's high sugars, then I know I've got to be careful. Why do I got high sugars? If I got high sugars beyond the optimum range, I got problems. I got to figure it out. Is there a water issue? Because sugars get diluted by the movement of moisture through the leaf, right? Water comes into the leaf, sugars get pushed out. So if I got high sugars, is there a problem with water moving through that leaf? Is there a stress factor? Is it too hot? Because you get to a certain point where respiration, transpiration slows down. I use what's called the 150 rule. I have it named after a dear colleague of mine who passed away last year. His name was Dell, and I call it the Dell rule 150. Now I apologize, it's Fahrenheit versus Celsius. See, Canada's messed up. We were metric and imperial at the same time because we live so close to the United States. I think liters per acre instead of liters per hectare. I'm a lost cause. So Fahrenheit, when you add the temperature as Fahrenheit to the humidity as a percent, the closer to 150 you get, the lower the leaf function. And that goes for pesticides, by the way, as well. Because if you're using a systemic pesticide, systemic needs to get in. If you're applying it and temperature and humidity combined are 130, 140, don't. Go to the contact. The systemics won't get in well because the leaf is starting to shut down. You got to apply that in the hours where the temperature and humidity are preferably under 100, ideally, but further away from that 150. So, and the same thing with fertility, because we need fertilizer to get in, it needs to be systemic. So we got to stay away from that 150. We want to be lower than that. So, but if I have high temperatures for a long period of time and it's not cooling down at night, the crop's going to slow down. The sugars are going to go up because we're not getting moisture movement. That's a function of the weather. We just need to be aware of it. It's a function of the weather. Insects can't digest complex sugars. And that's where the whole thing with the sugars. And what NovaCrop is looking to do next, by the way, is right now that sugar is a measurement of complex and simple sugars combined. We're going to start, NovaCrop is working now, one of the new things they're coming to is they're going to break it out. You're going to see simple sugars and complex sugars. So we can do an even better job predicting that insect and disease pressure. But if your sugars are low, you know that you're desirable for insects. Why are your sugars low? Something to do with photosynthesis and leaf function, right? That's where the sugars come from. So if we can get that leaf function up, get the sugars up, we're less desirable to the insects. Brix is kind of the big picture, but it may or may not be because of the EC not being broken out. That's why I prefer using the SAP versus just a little handheld Brix. Still good to look at that Brix reading. It gives you some indication, but this gives you the better picture. It gives you the whole picture. And by the way, SAP pH speaks towards the balance between the cations and anions. If the pH is high, then there's definitely either an anion imbalance, negative charge imbalance, or positive charge imbalance. You bring that balance into line, the pH will come into line. So the macrocations are balancing each other. The macroanions are pushing tug of war in with each other. The key micros are balancing and tug of war in with each other. I talked about silica. Nitrogen management. Here is one of the key reasons why I believe, and this is my last topic, by the way, and then we'll wrap up for lunch. Managing that nitrogen cycle. This is the one nutrient I think is the most abused, the most overapplied, and it's not cheap. But it's managing that nitrogen cycle. And by the way, that's what leads to botrytis. Dry oranges, granulation in citrus can be over application of nitrogen. There's a lot of disease and insect pressures that come into play because we're not managing the nitrogen cycle. And here's what I mean by the nitrogen cycle. There's two cycles. We all talk about the one in the ground, which was great. I love it, right? We talk about the monocle converting to nitrate. But when it comes back into the plant, there's another cycle. It's got to convert back from nitrate in the plant to a monocle before the plant can metabolize it into amino acids and proteins. And we need to manage that cycle. And that's where the moly comes into play. Nitrate to monocle in the plant requires a reductase enzyme that is moly dependent. If you don't have enough moly, that nitrate is just going to build up in the plant, and we're going to get insects and disease. And all what we need to do is put on a little bit of moly, and the nitrates get metabolized, and the insects will go to the other crop next door to the neighbor. Yes?
So what about adding amino acids as a source?
Oh, that's a big trend right now in North America, and it's a very good trend. It's expensive. But certainly at key times in the year, putting on an amino acid form of nitrogen is excellent. I'm a strong supporter of that. But the only thing we've got to be aware of, I mentioned it earlier, and you probably already are, is in a period of stress, those amino acids can still break down. So there's protein breakdown as well. But I like it because it's an amino acid is in a form that the plant is ready to use in a building block. So we see. Sorry?
How do you take it out of stress?
Take it out of
Stress.
stress. Depends on what the stress is. But potentially, well, you're creating, let's just say, you may not be able to take it out of the stress, but I will agree, we're creating less stress. because there's less energy required for the plant to utilize it. So we're feeding it the nitrogen and the building block it needs, and it requires less energy to utilize it. So yes. And it's a big trend right now right across North America to put on foliar amino acids, and foliar amino acid type nitrogen products tend to get in quite well. And basically they are either animal-based proteins that have been hydrolyzed, which means broken down into amino acids, or plant-based. Some people feel the plant-based ones are more natural and better. Others say, because the animal protein hydrolysis is cheaper. In the work that I've seen, and I haven't done any specific research, I'll say that up front, but on what I've seen, there doesn't seem to be a difference from what I've seen from plant response. But both have a role to play and are pretty neat. And certainly if you have it available, and if it's part of your program, it's probably helpful. I'm a big fan of that.
Phosphoric acids?
Phosphoric?
Acids.
Yes?
If you put it through the drip system,
Yes?
then when I was not farming organically, I was doing it.
Yep.
And you can definitely see the change in the plants.
Much better. Yes. First off, the pH,
Mm.
and I prefer phosphoric over sulfuric. Now here's something that I can't fully explain, but I'm going to share it anyways, even though I don't like doing it because I like to explain the why. But you brought up, and I think it'd be neat. I used to do a lot of work in greenhouse production. So greenhouse cucumbers, peppers, and tomatoes. And high pH water, 7.8, so we had to buffer it down. When we used sulfuric acid, over the course of a couple months, the crop would not look as well. We'd flip to phosphoric acid, and the crop would improve dramatically. Now, phosphorus levels would climb quite high, and we'd have to manage that at some level. But the crops seem to do better on phosphoric versus sulfuric or nitric. So I'm a big fan. If you're buffering your pH, phosphoric is the way to go. And by the way, phosphoric acid is orthophosphorus. We talked earlier about ortho being very available, so it's the better form of phosphorus. So it's a double win. So phosphoric is definitely a good product to use. And I prefer that over sulfuric or nitric. You got it. Big time. Because that's a huge problem with emitters, right? Clogged emitters.
You see, there's certain times that you can give amino acids for the plants.
Mm-hmm.
Which are the windows of opportunity?
Good question. Anytime before later season, once you start seeing color, stop. Because just like seaweed's an ethylene inhibitor, getting an excessive amount, whether it's an amino acid N or conventional N is an ethylene prevents color. So in the early season, so where I've seen the work is during that fruit cell division, after petal fall and before the end of fruit cell division. That seems to be a good window because that nitrogen is going to support the phosphorus and calcium in cell division, and the more cells that we can divide and create, the better the fruit's going to be later. So that's the window where guys will focus on putting their, in North America anyways, that's the window they'll put on the amino acid nitrogen, is petal fall to that end of fruit cell division. That's the optimum window of opportunity. Because you're getting the nitrogen that the plant needs so desperately for fruit cell division and fruit formation, cell formation, without having to expend a whole pile of energy to metabolize it. And by the way, I'll also say our simple sugars, the dextrose, lactose, calcium, and all that stuff, as simple sugars are also easy for the plant to metabolize because it's used to working in simple sugars. It doesn't have to convert it over to it's ready to use. That's why it can be metabolized and absorbed and metabolized within 2 to 3 days.
I want to come back to each phosphoric acid.
Mm-hmm.
When you do your SAP analysis, your phosphate is too high.
Mm-hmm.
You're going to have a deficiency of
Iron. That's what I mean by management, yes.
Before you do it, look at your SAP analysis before you do it.
And know if your phosphorus is getting too high, you're going to see iron deficiency. So put some iron on to prevent it. That's one of the drawbacks, is high phosphorus can inhibit micronutrient uptake. The first one impacted is iron, then zinc and manganese and copper. But the first one impacted is iron, so prevent it. If the phosphoric acid is what you need, and your phosphorus levels go up, manage it. If you don't need it, great, but if you need it as the buffer, that's the one to go to. As I said, I find the crops do the best on the phosphoric. but you do need to manage it. That's what I mean by managing it.
Minimum your iron, zinc, and
Iron, zinc, copper, manganese.
Microelements.
Yeah. Yeah, because those are critical. Formation and nitrogen metabolism, and the other ones are photosynthetic nutrients, right?
If you have an excess phosphate due to the phosphoric acid that you use, the way to get the iron, is it Phosphorous or can you put it in the ground?
Thank you, foliar. Foliar is the best. And easiest to move, by the way. We have our iron DL, and I'm sure there's other, but don't use the iron chelate, because chelates don't penetrate. You gotta use, whether it's ours or someone else's, use a product that was designed for foliar uptake. And it's, even with, it's amazing what 2 to 3 applications of 2.5 liters per hectare will move that needle against the phosphorus if you foliar apply it. It's harder to come into the root system because you already got the high phosphorus. The plant will struggle to take it up if you put it through the drip. You'll still get somewhere, you'll still move the needle, but if you want to move the needle best bang for buck, put it on the leaf. But using a form that's going to get into the leaf. It's just because you can use all it, or it's a liquid, doesn't mean it's meant for foliar application. So when we see in the SAP test, you can see you got the ammonium, you got the nitrate. This N in nitrate is a conversion factor. It's not a new part of the test. So they're just taking the oxygen away from the nitrate. They're looking at pure unoxidized nitrogen. And the reason they do that is because when the N in nitrate coming from nitrate approaches more than 50% of the total N, we know we're coming towards disease and insect pressure. So that's part of with the sugars. If you've got low sugars and a lot of nitrate and a lot of the total N coming from nitrate, we know if you don't have insects or disease, you're going to get it. So we need to get the nitrates down. Sulphur will do that, right? We know we need to get the sugars up. Photosynthetic nutrients, what's missing? Is it iron? Is it magnesium? Is it manganese? Is it a combination micro package? How can we get those leaves functioning to get the sugars up? Because if we get the sugars up and the nitrates down, those thrips and insects will depart. They won't be so happy. So that total N, this is what that total N represents. So you got your nitrates and your ammonium. There is a little bit of urea in there, but most urea doesn't come into the plant as urea, right? You get a little bit, but very little. So, but it is there. And then it's the nitrogen present as amino acids and proteins. So that's where that amino acid foliar is a good choice. And you will see it in your sap. I quite like it. And then here's the number, here's the magic goal. And by the way, just the last few slides, I put in some pictures that maybe don't fit the slides as well, but they're pictures of Canada that I wanted to share. This is one of the oldest wine grape plantings in Canada. It was planted in the late 1800s. Canada's a very young country. We just became a country in the late 1800s. So this is one of the oldest plantings. There's only one row left. It's, I believe it's a Muscat variety, a New York Muscat variety. And Grandpa planted it. It's now 3rd generation farmed on the vineyard, probably one of the best. And they primarily do whites because they're in eastern Canada where they don't get the heat units for a red. You just can't build the bricks. So they do a white and they turn it into a white bubbly. They have a specialty market that they're now getting recognized for globally as a specialty white bubbly. There's only one row. They've left one row of Grandpa's planting because they just can't take it out because it doesn't really produce very well. Correct. Exactly. So they left one row. You can just, I look at those vines. I look at the bottom end of those vines and gnarly vines, and if they could speak, it would be an interesting story, I'm sure. So they left one row. I talked about moly, so I'm not going to harp on that. It is often a forgotten nutrient. This is a large apple grower I referred to earlier. Originally, he gave me his toughest field of apples. The variety was a variety called Honeycrisp that has calcium deficiencies that creates a problem called bitter pit, very similar to blossom end rot on tomatoes. And he gave me his toughest block, and he said, "You do your sap." We did 4 saps through the season. You do your sap, and I'll put your products on, and we'll see where it goes. Now, yeah, I like to think I'm biased. We got good products, but the sap told us that we made some big changes. We took the potassium out of the program. He was having excessive potassium at the wrong time. We literally were able in his fields to take potassium out of the program. He had really low iron because his phosphorus levels were over 1200 ppm. Available phosphorus coming out of the ground. In SAP, it was way off. So we started doing full air iron. At the end of the year, he texted me this picture with a note. You turned my worst producing field into my best producing field. Now we just got to do it for the rest. And that's what we're now doing. We're turning them around as well. So that's what I'm looking for, is you as a farmer to succeed. I'm not just looking to sell you something. I want to give you the knowledge and the understanding of why to ask, so that you can succeed. And then I want to see pictures like this that tell me that your crops are the best that they've ever been. That's it. And there's a picture from 2 years ago. That is an apple tree in central Canada. And as you can see by the apple tree, if you look closely, there's flowers. It was, for us, June is coming into the early summer. This picture was taken early June. We had 10 centimeters of snow. It went down to -3 for 6 hours. Very little. The crop was completely wiped out in this area. It was devastating for those farmers. And unfortunately, when you lose a crop, apples are a crop where next year's buds are setting as this year's petal fall is happening on some varieties. So not only, and then the apples that developed, their seeds didn't develop as well. They had immature seeds. And as seed develops in the crop, it gives off hormones, right? That help with the development process. Well, because the seeds didn't develop properly, the hormonal balances were wrong. So the subsequent year also had horrible yield. It's now 3 years later, and even this year isn't right. It's very uneven set because of that. So when a crop gets out of balance, whether it's grapes or apples, it can be detrimental. So we want to do our best to avoid that, but sometimes Mother Nature steps in, and you just got to say, ouch, and move on. So anyways, thank you. I hope you enjoyed it. Sorry that I talked for so long, but I hope you found a few nuggets. I'm here over the lunch. If you have any questions or something you want to talk to me about one-on-one, please come talk to me. And thank you for your time. It's a pleasure to be here. I'm honored to have your morning, and you have a beautiful country, South Africa. It's my first experience in South Africa, and it is amazing. Thank you.
Thank you.